Hi everyone, my name is Yunsu and Xiaoqi and I will present our project. In this project, we attempted to predict chromatin accessibility from DNA methylation probability. From the paper, The Chromatin Accessibility Landscape of Primary Human Cancers, we were able to download two datasets. The first is the ATAC-seq data. ATAC-seq is a technique used in molecular biology to assess genome-wide chromatin accessibility. The second data we downloaded is the DNA methylation data. DNA methylation is the biological process by which methyl groups are added to the DNA molecule. After downloading our two main datasets, we also downloaded the ID/gene mapping data. Using the gene mapping information, we were able to do a one-to-one mapping between ATAC-seq data and DNA methylation data. This part was really difficult because we didn't understand the data well in the beginning, and also because we had to do the mapping ourselves to create our training and test datasets. After mapping the two datasets, we constructed our training data. We have several features in our training data. First is the methylation information, which corresponds to the X value in the table, chromosome number, and chromosome range, start and end location. And the ATAC-seq data is our Y labels. In total, we were able to obtain about 5 million data points, and we split 20% into test data.
As for the model part, we firstly created a CNN as a baseline model, which generates local receptive fields to learn the input data. As for a CNN model, the most crucial hyperparameters are kernel size and layers. Therefore, we applied CNN with different depths and widths. The structure of the best CNN model to be achieved is shown here. Our CNN model is consisted of two convolutional layers, which followed by a bed norm layer, a drop layer, a multipling layer, and an ILU layer. After the convolutional network, we added a dense net with two linear layers. And in addition, we constructed a recurrent neural network. RNN is different from CNN because RNN considers sequence information, whereas CNN only takes into account of the spatial distribution of input. RNN can be regarded as a neural network transmitting in time. Since we know that DNA data share sequence information, it is natural to try applying an RNN model. Aside from sequence information, we also want our model to maintain the ability of dealing spatial information. Hence, based on the model we designed previously, we added CNN layers after the RNN layers. This way, we consider both sequence and spatial information. As shown here, our model is consisted of two RNN layers, followed by four COM1D layers, each with a batch normalization, a drop plot, a multiply, and an ILU layer. Then we added two linear layers at the end of the model. Here are some results. Our code is uploaded on GitHub and can be found in the link below the slide. We conducted experiments with different number of layers in each model. Our CNN plus 1 RNN model with 8 layers performed the best of all the models. We assessed our results using accuracy and loss, and we used binary cross-entropy to measure the loss. Our best model used 0.898 test accuracy and 0.431 test loss. The second best model was CNN plus RNN with its layers, which yield test accuracy of 0.856 and test loss of 0.472. The RNN model by itself did not perform as well as the CNN. However, when we combined the two, the performance was the best. Thus, this result confirmed our hypothesis that combining CNN and RNN would perform well, as the model considers both short and long-range information. Looking at the sequence information as well as individual details, in terms of experimenting with different number of layers, having more layers generally produce better results, which was as expected. Here are some discussions of our whole final project. The most difficult aspect in our project was data processing, and we spent a lot of time in this part. We not only needed to process the data, but also understand the relationship between each feature. It was extremely difficult to map multiple datasets that we had to one another. In order to improve the model, understanding the model is very crucial. This was a challenging for both of us since we both do not have a genomic background. As for some future work, we only focused on breast cancer data, but it would be interesting to reproduce this work in other cancer data as well. Moreover, in terms of improving the data, since DME and ATSSR both have spatial-wise and sequence-wise information, we could try applying some kind of attention mechanism. Attention mechanism could allow us to focus on long-range information. So here's the end of our final projects.